Ok, ahora sí. Ok. Bueno, buenas tardes. Bienvenidos a todos al último seminario institucional del mes patrio. Es para mí un placer presentarles al doctor Jason Torres, líder del Departamento de Genética Epidemiológica de la Universidad de Oxford. Él es originario de Texas, donde estudió biología. Su interés en metabolismo lo llevó a realizar su PhD en la Universidad de Chicago con la doctora Nancy Cox, que es una pionera en estudios genéticos con asociación a enfermedades. Después de su doctorado, se unió al grupo del Dr. McCarthy en Oxford, donde se dedicó a analizar regulación génica epidemiológica enfocada en diabetes. Hoy nos viene a contar sobre uno de sus principales proyectos, el estudio prospectivo de la Ciudad de México, donde colabora con la Facultad de Medicina de la UNAM y nos viene a contar el proyecto y lo que han hecho. Entonces, sin más preámbulos, escuchemos hoy qué gritan nuestros genes, además de Viva México. Y Jason, thank you very much for being here, and they are all yours.
Okay, check, hello. Gracias, Jaime. Es un gran placer. Estoy aquí con ustedes. Thank you very much for being here today. It's a great pleasure. I've been looking forward to this for a good few weeks now. I've been excited to come to Mexico again, partly because I miss the sun, although I do feel bad because I seem to have brought earlier this week the terrible British weather with the clouds and the rain, but it seems that England is wearing off me now. So as Jaime mentioned, my name is Jason Torres. I am a genetic epidemiologist working at the University of Oxford in the Department of Population Health. And today I will share insights from the Mexico City Prospective Study. So for those who may not be familiar with MCPS, this was a study that was initiated over 20 years ago by two epidemiologists working at UNAM in Mexico City, Professor Roberto Tapia Cañar and Professor Pablo Curi Morales. And 20 years ago, they were interested in studying the impact of smoking on health in Mexico. At that time, it was understood that smoking was a leading cause of premature death, but most of those studies were done in the United Kingdom and the United States, and the extent to which smoking impacted population health in Mexico was unclear. In fact, there were very few studies outside of the US and the UK at that time really looking at smoking. So Roberto reached out to epidemiologists working at the University of Oxford, including Sir Richard Dahl, who in 1950 made the initial discovery linking smoking to lung cancer. And this contact eventually evolved not just into a series of conversations, but a collaboration with the plan to develop a study that would not only look at smoking in Mexico, but other potential causes of disease and premature death. So this was the beginning of the Mexico City Prospective Study, or MCPS, which is now the largest blood-based prospective study in Latin America. Between 1998 and 2004, over 150,000 adults were recruited from two districts in Mexico City, the centrally located district of Coyoacán and the adjacent district of Iztapalapa. At baseline, a team of field workers, including registered nurses, administered a questionnaire, took physical measurements, and importantly extracted a 10 milliliter blood sample from each participant. Between 2015 and 2019, a resurvey was conducted in a subset of 10,000 participants, and notably, there is linkage to a national death registry, which keeps up-to-date records on mortality with information on primary and contributing causes of death. Now, if this study were done today, the questionnaire would likely be administered using some type of app on a phone or an iPad, but back in the late 90s, they used a barcode reader. This is what paperless data collection looked back then. And a question on the questionnaire would have been facilitated through this reading of barcodes. So on the right, we have an example of a question that participants would have received concerning alcohol consumption. So in the last 12 months, how often did you drink any beverage with alcohol, such as beer, pulque, wine, etc.? You can see there are different options, such as daily, 2 to 3 times per month, with the corresponding barcode. So this was used to administer the questionnaire, which turned out to be quite accurate and well calibrated. And at baseline, a whole range of questions were taken. Information about age and sex, occupation, income, health service provider, all was ascertained from each participant. Information on lifestyle characteristics, diet, smoking, alcohol consumption, and importantly, the field workers asked about previous diagnosis of diseases, as well as any medications that participants were on. In women, information on reproductive history was ascertained, and notably, physical measurements were taken from each participant: height, weight, waist circumference, hip circumference, blood pressure. And a 10 milliliter blood sample was extracted, which was used to collect plasma and buffy coat, as well as conduct chemical assays such as measuring glycated hemoglobin, HbA1c, other assays, and more recently, NMR-based metabolomic profiling for over 200 measures based on nuclear magnetic resonance. So a lot of information has been taken for MCPS, and if you're curious about what data is available, there is an online data showcase. So if there's a particular phenotype you're interested in studying, you can go to this website now and see if information on this was ascertained either in the questionnaire or at resurvey or in the mortality records. Now, one of the most important features that distinguishes MCPS from other large prospective studies conducted in the US and the UK and other global populations is the high rates of obesity and diabetes in this cohort. At recruitment, by age 50, 1/2 of all women and 1/3 of all men were obese, with the BMI greater than or equal to 30, and 1 out of 5, 20%, 20% of participants had a previous diagnosis of diabetes. And for context, this is more than double the rates observed in the US and the UK at that time. And in fact, the leading cause of premature death in Mexico is not smoking, it was found to be diabetes. So among all adults aged 35 to 84, a previous diagnosis of diabetes more than quadrupled your risk of dying, increased the risk of death by a factor of 4, corresponding to a death rate ratio of over 4, and was highest among adults aged 35 to 59, where a previous diagnosis of diabetes corresponded to a death rate ratio of 5.4, more than 5 times likely to die. And in fact, it's estimated that 1/3 of all deaths in this study among adults aged 35 to 74 was directly attributable to diabetes, with a dramatic increase in mortality resulting from a previous diagnosis of diabetes, particularly when it comes to kidney disease, cardiac disease, and infectious diseases. So since then, there have been a number of other studies conducted in MCPS resolving the genetic, or not the genetic, but the key risk factors for disease and premature death in Mexico, such as smoking, which of course is a major risk factor, diabetes, adiposity, alcohol intake. We just had a paper accepted last week looking at alcohol and mortality and cause-specific mortality published in Lancet Public Health. But most of these results are based on observational epidemiology. But this talk is a genetic talk. And so I'd like to talk about what we've learned from genetic analysis in recent years in this cohort. So if you're familiar with the contemporary state of human genetics, you've probably seen a figure like this many times before. There have been many genetic studies conducted in the past 15 years, specifically using a method called genome-wide association studies, or GWAS, which are these large comprehensive tests that involve thousands of individuals testing millions of markers. But the vast majority of these GWAS studies have been done in populations of European descent. So if you look at the current GWAS diversity monitor, which keeps an up-to-date record of the total participants in GWAS studies, as of last week, over 93% of participants are of European descent, whereas less than 1% of participants are of Hispanic or Latin American ancestry. And you can see here on the right that this trend has not improved in the past 10 years. So there's still a stark disparity in the representation. And this is not just a problem for representation's sake. There is important information about genetic variation present in Latin America that can be quite informative. There are unique patterns of variation in Latin America that have been shaped by a complex history, demographic events involving the migration of indigenous communities throughout Mexico with subsequent differentiation and population isolation events. And then in the 15th century, you had European colonization, right? From Spain and Portugal coming into the Americas, introducing European genetic variants in this population. And this is a very interesting point to stress being here in Cuernavaca today, because we're only a few miles from El Palacio de Cortés, right? Which was kind of the epicenter of this massive consequential colonization event of Latin America. And then since then, there were epidemics, infectious diseases that caused population bottlenecks and then subsequent expansion. So all of these demographic events have shaped variation in Mexico, resulting in unique patterns of variation that could be informative when we look at phenotypes and diseases. So this was something that Roberto and Pablo were aware of over 20 years ago. But at that time, the Human Genome Project had just finished. We, for the first time, had a complete sequence of the reference, but we were many, many years away from having cost-effective technology that could genotype and sequence DNA. But he did seek informed consent, and that was a major reason for collecting blood from participants. And in fact, it wasn't until 2020 that genetic data finally became available in MCPS through a partnership with AstraZeneca and Regeneron, specifically the Regeneron Genetics Center, which has now made it available to have access to genome-wide genotyping using the Illumina Global Screening Array V2, which has been conducted in over 140,000 participants in Mexico City. And exome sequencing has been done in over 140,000 participants. Again, this is the region of the genome that encodes proteins. And importantly, we also have whole genome sequencing data available for a subset of 10,000 participants of who are mostly unrelated. So not only is MCPS the largest blood-based prospective study in Latin America, it's now one of the largest genetic studies in any non-European population. So this is when I joined the project, which was a good time to join with the data being available. And we wanted to characterize genetic diversity in MCPS to identify features and patterns of variation present in this population. And when Jesus Alegre Diaz, who led the fieldwork team to recruit participants, when he did that study, he and his team actually knocked on 100,000 doors in Coyoacán and Iztapalapa, right? They just went up to people's houses and recruited as many people as were willing to consent. So you often had multiple family members within the same household participating in MCPS. So we knew there were a lot of families in MCPS, but the extent to which people were related was unclear. So that was the first thing we did, is we characterized relationships. And we did this using a method called IBD, estimation identical by descent sharing. So identifying regions of the DNA between people that were inherited from a common relative. And using IBD segments, we could infer the overall level of relationship present between people. And from this analysis, we found over 31,000 parent-offspring pairs, over 29,000 full sibling pairs, and notably, at least, we found that 71% of participants had at least one close relative within the third degree, so first cousins, primos, or closer, right? So a lot of family relationships present in this cohort. And in fact, if we take these IBD estimates and build networks of shared first-degree relatives, we find that these become quite extensive. So we reconstructed over 22,000 first-degree networks of shared first-degree relatives, such as parent-offspring or full siblings, identified these networks that had over 65,000 individuals and nearly 3,600 nuclear families. The largest first-degree family network is shown here, with 48 participants, right, from the same family in this study. And it becomes more complex when we allow for second-degree relationships. So this plot here has shared second-degree relationships where you have at least 4 people in the same family network, where each circle or node is a person, and each line or edge is a relationship. And so you can see that it becomes quite extensive and complicado, right, with all these relationships. The largest relationship network had over 9,000 participants with shared second-degree relationships binding them together. So this is something we didn't expect. It was quite extensive. But this is not terribly surprising in the sense that a lot of people within the same extended family often live proximally together, and that's what we found out, is that many people, especially in East Iztapalapa, have extended relatives living close by. So the key thing to take away, first result, is that there are many relationships present in this MCPS cohort. Now, because we had exome sequencing and whole genome sequencing data present, we wanted to compare variation to find out what type of variants, SNPs, single nucleotide polymorphisms, insertion, deletion, structural variants in MCPS. And for those in human genetics, this is an exciting time to study DNA because there are now large genetic data sets available. Many of them are freely available and accessible, such as the UK Biobank, right? So this is another project. This project is led by researchers in my department, and it includes over 500,000 participants from the United Kingdom. And currently, researchers could access 470,000 exome sequences. And now, just since last November, so less than a year ago, there is whole genome sequencing data available for 500,000 participants. And researchers globally could apply to access and analyze this data now. But there's also databases such as the Genome Aggregation Database, or GNOMAD, which has exomes for over 730,000 participants and over 76,000 whole genome sequences. And the TOP-MED dataset, the Transomics for Precision Medicine cohort, which has whole genome sequences for over 132,000. So there's a lot of data here, but we wanted to see how genetic variation in Mexico compared with these datasets, which are largely based on European descent individuals. And to give you some brief summaries, when we analyzed the exome data for 140,000 participants in Mexico City, we found that coverage was good, so the quality of the data was such that 98.7% of samples had 90% of the bases with a high coverage of more than 20x, so pretty good read depth. We called variants using the OQFE workflow and applied a machine learning-based quality control step to identify over 9.3 million exome variants present in MCPS. Of these 9.3 million variants, 4 million were coding variants present in the exons of genes. The protein encoding components for these 19,110 genes, and the vast majority of these variants were rare, less than 1% frequent in this population. Of these 4 million variants, 233,000 or so are what are known as predicted loss of function variants. So these are genetic mutations that introduce frame shift, alterations, or stop gain mutations that cause premature truncation of the protein. So they severely compromise the structural integrity and the function of protein. So these are very damaging mutations present in MCPS. And when we compare MCPS, this Mexico data, with data from the UK Biobank dataset and GNOMAD, we find that 1.4, more than a fourth, of these variants are unique to MCPS, that they were detected in this Mexico population for the first time. There is a lower proportion of singletons, so these are mutations that only occur in a single person. There was a lower proportion of these singletons in MCPS compared to the UK Biobank, but we had a higher proportion of homozygous predicted loss of function variants, where a person has 2 copies of these deleterious mutations. And both of these observations are due to the fact that we have many families, right? As we saw, there are many relationships, so that affects the number of variants present, of such variants present among individuals. We also looked at whole genome sequencing data, which again is available for about 10,000 participants, 9,950 of which are unrelated with a mean sequencing depth of 38.5. And in total, there were 131.5 million variants in MCPS. 1.5 million occurred in coding regions, but in total, 31.5 million variants were unique to Mexico, right? Not present in UK Biobank or GNOMAD or TOP-MED. So that was quite interesting to find. So the second thing to point out is that there are unique rare coding and non-coding variants present in this Mexican population. So the third thing I'll move on to is an interest in characterizing population structure. Right? So population structure is a phenomenon where within a population you have subpopulations with systematic differences in allele frequencies, likely reflecting different ancestries present in the data. So one way to estimate this, or to characterize this, is to do what's known as a principal component analysis. Sorry, let me play this. Uh-oh. There we go. So principal components analysis is a mathematical procedure where you take a high-dimensional data set and you decompose that into what are known as principal components, which summarize axes of variation. And when you do this with genetic data, you often find that this distinguishes individuals based on geography and underlying shared ancestry. So if we do a principal components analysis using reference samples from West Africa, Nigeria and Yoruba, with individuals from Spain, Iberian samples shown in blue, and a subset of indigenous American samples shown in green, you find that this does distinguish them in this PC space, where individuals do cluster differently depending on where they were ascertained from geographically. So there's population structure here, right, representing differences in genetic frequencies. Now, if we project 10,000 unrelated individuals from Mexico City into this plot, you see that rather than clustering in a particular tight cluster, they map along this axis, right? Where you can see that some individuals fall closer to this green cluster, having more similarity with indigenous American participants, and a few having mapping closer to this European cluster here. But most individuals fall along this line, right? Having somewhere in between. This is known as genetic admixture, and it's well understood that Latin Americans, and especially those in Mexico, have a high level of such admixture. So we can see that there's, distinct population structure separating those with more European, indigenous, and to a lesser extent African ancestry, but we wanted to explore this further. So we collaborated with Professora Lorena Orozco, who leads a research group at INMEGEN, the National Genomic Medicine Institute in Mexico City. And for the past 12 years, Lorena and her team have traveled throughout Mexico and have been recruiting indigenous participants from different regions in the north and then in the south. So the northern desert Arido American regions, and then the temperate and tropical Mesoamerican regions, right, which historically were the sites of the Aztec Empire, the Mayan empires, the Olmecs. And what she found, and her team found when they characterized genetic variation among these indigenous participants, is that individuals largely separated based on their geographic regions. So indigenous people from the Yucatan and Campeche and Chiapas had more genetic similarity with themselves than between those in the central or southern regions, as you can see here. So they found that there were broadly 5 similarity groups based on the geographic regions. So we worked with Lorena and used genetic information from MACE, this metabolic analysis of an indigenous sample, to characterize genetic variation present in Mexico City. And when we run this PCA analysis and include the MCPS shown in black with the MACE samples shown in these different colors here, among the MCPS participants that have more indigenous ancestry, they tend to coincide with indigenous individuals from the central, southern parts of Mexico. So the area right around Mexico City, and then the southern regions of coinciding with Oaxaca and Veracruz, and to a lesser extent, Maya from Yucatan. This was based on PCA analysis of individual level genotypes, but for those familiar with haplotypes, which reflects the fact that variants are shared together, that you inherit multiple variants correlated with each other from our parents, you can use haplotype information to generate more fine-scale structure. And when we generate characterized population structure using haplotype information, we can see that there are these discrete arms or regions within this PC space that correspond to distinct ancestry groups. So this arm here on the upper left has the highest level of Southern European ancestry corresponding from likely Spanish ancestral population. This arm here, this purple one, largely has the highest level of similarity with indigenous groups from the southeastern part of Mexico, kind of overlapping the Maya linguistic speaking people of southeastern Mexico. This arm here shows high level of similarity with indigenous groups from the central part of Mexico, outside of Mexico City, including Morelos. And there were actually two subpopulations representing indigenous groups from the southern part of Mexico, largely coinciding with the Otomangue speaking linguistic people there. So this is based on using principal components analysis, which is a unstructured genetic method, but we can use parametric analyses to estimate what are known as individual level ancestry proportions. Sometimes the word people use is global ancestry. And when we do this, we can, for a given individual, estimate the total amount of their genome that are inherited from different ancestral populations. So here I have reference samples from Africa or African background, East Asia, and Europe. So each vertical line is a person, and each color is an inferred ancestry. So if we, let's say, look at the European component here, the light blue is highest among Northern Europeans, where Finn stands for Finland. We have the highest blue ancestry group among the Finnish, whereas the dark blue is highest among Southern Europeans. So IPS would be Iberian from Spain, España, and TSI would be Italian from Tuscany. So there are two ancestries we can characterize in Europe in this data set here. This is what the results look like when we include indigenous populations from Mexico. There's a lot of diversity, right, in this population. It's very impressive. You have broadly these brown color, this brown ancestry group, which is highest in the north, right, very high among the Tarahumara, the Guarajillo. But then you have high level of differentiation among the city, right, which is an indigenous group that lives in Isla Tiburon in Baja California. It's literally on an island, so there's more differentiation. So there's a high level of diversity there. The purple is highest among the southeast, among the Maya linguistic families. The pink is highest in the south, and this tan is highest in the central region. So there's an incredible population structure present among indigenous communities throughout Mexico. This is what the results look like when we include nearly 140,000 participants from Mexico City. So you can see here, again, extensive admixture, with some individuals having more dark blue, which is Spanish, right, likely or Southern European, and more individuals having higher level of indigenous American ancestry, particularly from the south and central parts of Mexico, with the average global ancestry proportion being around 67% indigenous in Mexico. So a highly admixed population. Now this is what's called global ancestry, but we understand that for a given Mexican admixed participant, that their genome is a mosaic. Some parts of the genome are indigenous, some parts of the genome are European, right? And this is what's known as local ancestry. So the next thing we wanted to do is characterize this local ancestry features in MCPS. So there's a method called local ancestry inference, and one method that's widely used is called RFMix. It's a software program for inferring local ancestry. So the way it works is you have reference panels, reference individuals that represent different ancestral populations. So if we imagine somebody who's African American, right? An African American has ancestry from both European and African background. So if, let's say, blue is European and red is African, you would have phased DNA, phased haplotypes from both a European and an African reference population, you divide the genome into these loci, these DNA segments, and within each segment you observe the pattern of genetic variation. Are you guys familiar with SNPs, single nucleotide polymorphisms? So these are single nucleotide variants, and so you could genotype them within these haplotypes. And then here you have in gray an admixed chromosome. So this could be the DNA of an African American participant. And the way the program works is you take the observed data and you train a mathematical model. In this case, it's a model known as a random forest. This is a set of decision trees. Each of these decision trees has a random number of the SNPs used to develop a classifier, so that each tree predicts what the ancestry is. So red would be more African, blue would be more European. And so each tree makes a prediction, and you average the predictions across the trees, resulting in a overall inferred ancestry for this individual. So here, this is the observed DNA in gray for the admixed chromosome. And when you apply the model using the reference data, you basically infer that up to this point, the individual is African, and then from beyond, it's European. So this was the method we used to characterize local ancestry in Mexico, but we used a three-way model, allowing for African, European, and indigenous. And when we did this analysis, we found that there indeed were complex patterns of variation within people. So on the left here is a karyogram showing the local ancestry for the 22 autosomes, right? The non-sex chromosomes. And so you can see this person here has mostly African ancestry with some indigenous admixture, whereas this person here has extensive admixture from both European and indigenous American ancestral population. So there's a ton of diversity that exists among participants in Mexico. And if we average these local ancestry segments across the genome, we find that they're consistent with the global ancestry estimates. So I mentioned a moment ago that when we look at the global ancestry proportions, there's about 67% indigenous per person. And so when we average the local ancestry, we see that across the genome, for most autosomes, it's about 60, 66, 67%. Indigenous, about 30%, 33% European, and then a small fraction, less than 3% African, right? Ancestry across the genome. But there were some differences, right? So here's chromosome 6. And as you can see at chromosome 6, there is the locus where there is a decrease in indigenous and a decrease in European, but an increase in African ancestry. This is the HLA or MHC locus, which plays an important role in immunity. And it's thought that because of the greater genetic diversity among Africans, that this might have had a selective advantage that was protective against infectious diseases. So this is one hypothesis. Another feature is that when we look at chromosome X, we have higher indigenous ancestry on chromosome X than among autosomes. And this is something that's been characterized before. It's called sex-biased gene flow, which reflects the fact that when the Europeans came to the Americas, right, as a consequence of colonization, it was more likely for European men to pair with indigenous women than the other way around. So again, this is a genetic feature of colonization, right? So it's really stark to see this. But the fourth take-home message, right, or third take-home message is that there is extensive admixture and Mesoamerican ancestry present in MCPS. So with this in mind, we can now combine data, right? We looked at the relationships, we looked at the genetic variation, and we looked at the ancestry. So you can start to put this data together to infer things about the variants. So for example, if we take the sequencing data from the exome and whole genome data sets, and we combine it with the local ancestry segments from the analysis I just shared, we can estimate the ancestry of each variant using the local ancestry segments and the exome data together, if they were given that they were phased aligned on the same reference scaffold. And with this data, we can do a few things. We can calculate using the probability of a given ancestry for a variant in an individual. We can get the weighted allele number of ancestry K. So how many alleles come from, let's say, indigenous background. We can then take into account the genotype information and calculate a weighted allele count for that ancestry K. So for a given variant, for a given allele at a variant, find the number of counts of that from indigenous ancestry, divide that by the total number of alleles from that indigenous ancestry to calculate the allele frequency of that allele in that ancestral background, right? So estimating population-specific allele frequencies from this analysis. And what's interesting is that when we do this and we compare the inferred ancestry-specific allele frequencies in this admixed Mexican population with actual allele frequencies from the nomad data set, we see there is a high level of concordance. So if we look here, for example, on the far left, we have non-Finnish Europeans, so actual European individuals from the nomad data set, and then we have the allele frequencies estimated in Mexico, just looking at the European segments among this admixed population, and we compare these two values, we see that the R squared, the concordance is very high, right, 0.994. If we look at African, it's also high, 0.98. And that's impressive or interesting given that there's only 3% African ancestry on average in Mexico. But if we just isolate the African segments in the Mexican population, we can recapitulate allele frequencies. And then we see, though, when we look at the Latino admixed American component in nomad, with the indigenous American ancestry inferred in MCPS, it's not as high. And that's because the admixed data set in nomad has more European in it. So we're able to more precisely estimate indigenous allele frequencies when we use this approach. Now, when you calculate allele frequencies, this could be biased if you have close relatives in the data. And as I mentioned, we have a lot of relatives, right? Hay muchas familias in MCPS. So using that IBD information I mentioned earlier, we can correct for the allele frequencies. So we use this IBD graph approach, and the way it works is, let's say we have a variance where there's two alleles, 1 or 0, and we want to get the allele frequency of the 1 allele, right? Well, we can see here that there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? There are 10 haplotypes. And if we count the ones, there's 1, 2, 3, 4, 5. So 5 of those alleles, 10 haplotypes, 5 over 10, that's 0.5, right? That would be the observed allele frequency. But these three individuals here share a haplotype. They inherited that from a common relative. So that's actually one unique haplotype present in the data. Same thing here. These two individuals share that blue haplotype, right? This segment of DNA, I should say. They share this segment identical by descent from a common relative. So if we collapse those IBD networks, then there's really 1, 2, the whole triangle, that's just now 1. So it's 1, 2, 3, 4, 5, 6, 7. Right? So there's 7 unique IBD segments. And if we collapse the allele count in that triangle, there's now 1, 2, 3. So 3 out of 7. So instead of an observed allele frequency of 0.5, the actual allele frequency is 0.43. So we now have this more accurate information on ancestry-specific allele frequency when we leverage all of this data, right? The IBD, the sequencing, and that local ancestry. So we did a lot of work, and I won't have time to go into all of it, but we did imputation experiments. We looked at genetic prediction using polygenic scores and evaluated by ancestry. And then we investigated a phenomenon known as runs of homozygosity, these stretches of DNA that are homozygous. So we have a paper that's published where you can read about this. And it was published last year in the journal Nature. So it's this one on the bottom, genotyping, sequencing, and analysis of 140,000 adults from Mexico City. However, we were pleased that this paper was published alongside a separate paper led by researchers here in Mexico. There was this study called the Mexico Biobank, which is led by Professor Andres Moreno Estrada, who has a research group in Guanajuato. But the first author of this paper is Professor Mashal Sahal, who is an associate professor here in Cuernavaca. Right? So it was actually amazing that both of these studies were published together. And there's nice complementarity, because the Mexico City Prospective Study has many individuals and a lot of data from Mexico City, right, from Coyoacán and Iztapalapa, whereas Mexico Biobank has about 6,000 samples from people all over Mexico. So there's more diversity there. And I'm actually excited because now Mashal and I are going to start collaborating on a project on BMI, so on obesity. Now, we created a number of resources from the Mexico City Prospective Study project. So all of this local ancestry, this ancestry-specific allele frequency results I was discussing is publicly available. So there is a website you can go to now called the MCPS variant browser. So if there is a variant, a SNP, indel that you're interested in, you can go to this website and see how frequent is this variant among Europeans, among Africans, among indigenous American ancestral background. So here's an example where you see a variant that is common among indigenous Americans, about 51%, but well, yeah, it's common in all of them, actually. Sorry, it's not a great example. Also, you can analyze this data now. It's available for sharing. So we created, we have a platform on the DNA Nexus cloud server where researchers can access the data and analyze it. In fact, on Thursday last week, I co-led a workshop on analyzing genetic data using the DNA Nexus platform in Mexico City. And I recognize a couple people here from that workshop, so it's good to see you again. And I'll come back to data sharing in a bit, but this data is available for researchers and is exclusively available to researchers based in Mexico for a protected time. Now, I'm running out of time, Aime, but I would like to maybe spend another 5 minutes talking about some of the results we've learned about diseases in MCPS. And in particular, we have exome data, which characterizes rare variants in MCPS. And rare variants, specifically these predicted loss of function variants, could be informative, right? Mutations that damage the protein, that impair its structural integrity and its function, can be very useful in identifying relevant biology and potential drug targets. So there is a method called a collapsing rare variant burden test, where if you consider qualifying variants, that's what's called QV, qualifying variants, these that you care about, such as these predicted loss of function variants, you can then do disease-based association test where you test genes. So for a given gene and a given disease, you count the number of cases and controls that either have these protein predicted loss of function variants, these protein damaging variants. You look at the count of cases and controls with these variants among those with the disease, among those with the qualifying variants versus those without. And then with that information, you can do an association test to see which genes have an association with such damaging variants. And this has been quite informative in telling us about the biology of different phenotypes. So for a few years ago, we contributed to the largest rare variant study of obesity, looking at BMI, in a study of over 640,000 individuals, including 100,000 participants from MCPS. And we found that there were 16 genes where these rare loss of function variants associated with BMI. What was interesting is that one of these genes, GPR75, this G-coupled protein receptor expressed in the brain, hypothalamus of the brain, was such that rare loss of function mutations in this gene associated with lower weight. So this is the distribution of all of these protein truncating variants identified. And basically, the mean BMI among people with these mutations was lower than those without the mutations. Now, these are very rare mutations. Only 4 out of every 10,000 people in Mexico have them, or cross studies have them. But among those with these mutations, there was a 0.34 lower standard deviation, 0.34 standard deviations lower BMI, which corresponded to 1.8 kilograms per meter squared lower BMI and a 54% lower odds of obesity. So these are rare variants, but they were protective against obesity. We did studies in mice where we knocked down the gene using adding deletions, either heterozygote or homozygote deletions of the mouse homolog GPR75, and found that when you took these mice and you fed them a high-fat diet for 2 weeks, they all gained weight, but the mice that had deletions in this gene gained less weight. So those that had one deletion gained 25% less weight, but those that had complete knockout, 2 deletions of GPR75, had 44% less weight gain. And this corresponded to better glucose tolerance after 2 weeks. I'll wrap it up now. Okay, fair enough. We did some more analysis. So we did a follow-up study looking at a body fat distribution, looking at waist to hip ratio. And we found another gene, inhibin beta E, INHBE, where mutations, protein truncating mutations in this gene associated with a healthier metabolic profile, so lower waist-hip ratio, which was significant in Mexico. It was significant when we meta-analyzed it across studies, and this corresponded to a 28% lower odds of having diabetes. I mentioned the first exposure I talked about today was smoking. So last year, we contributed to a study of smoking that included rare variants from Mexico and other populations. And what we found were that rare loss-of-function mutations in the gene CHRNB2 reduces the likelihood of being a smoker. Right? This is a gene that encodes the beta-nicotine acetylcholine receptor expressed in the brain, which is involved in the drive, the pleasure you get from smoking. So individuals with mutations in this gene were less likely to be heavy smokers and was also, present in Mexico. The last thing I'll talk about, which I think is very interesting, is a rare variant study in diabetes. So we contributed to a study led by our colleagues at AstraZeneca to identify these rare protein truncating variants that associate with diabetes, and we again found a new gene, MAP3K15, a gene present on chromosome X, where rare loss-of-function variants in this gene associated with less risk of diabetes. So damaging mutations in this gene lowered your risk of developing diabetes. And this replicated in Mexico, right? So individuals in Mexico that had these rare loss-of-function mutations in MAP3K15 had a lower odds ratio for diabetes and lower anti-diabetic medication use. But this isn't the only gene that's involved in diabetes risk. There are many variants that influence risk for type 2 diabetes, in type 1 diabetes. And 10 years ago, the Sigma Consortium led a large sequencing study in Mexico. And they found a genetic association at the gene SLC16A11. This was the first time this mutation was reported, or the variants in this gene were reported, associated with diabetes. There were 5 variants that were associated with diabetes at this locus, and using this information, they identified there were 3 haplotypes, so sets of alleles that were inherited together. And the risk haplotype here, which had 4 missense variants, was common in Mexico. So among all participants, 30% had the risk haplotype in Mexico, and among those with mostly Native American ancestry, that was almost 50%. So 48% had this risk haplotype, whereas it was 12% in Asians, less than 2% in Europeans, and not present in Africans. So it's a risk variant that's very common in Mexico, and in addition to being common in Mexico, it has a high effect. The odds ratio is 1.3. So basically, each copy of the allele confers a 30% greater risk of diabetes. So we did a GWAS of type 2 diabetes in MCPS, and sure enough, one of our top associations is SLC16A11, this mutation that's common in Mexico. But if we look at SLC16A11 and we come back and look at that rare variant at MAP3K15, that protective mutation, we see that things are a bit different, right? So again, MAP3K15 is protective, and it lowers your risk for diabetes by about 30%. Whereas SLC16A11 is common and increases your risk for diabetes by about 30%. So if you have somebody without the risk allele, the risk haplotype at SLC16A11, but they have the protective variant at MAP3K15, they have lower odds of diabetes. But if they're part of the 30% of the population that has the SLC16A11 haplotype, there's no protective effect on overall. So this underscores the importance of considering the unique genetic background of each person in how they respond to, how they may respond to intervention, and underscores the importance of personalized medicine, has implications for personalized medicine. So I'll wrap it up. MCPS, it's the largest genetic study of a Latin American population. There, as we saw, many families, complex ancestry, and admixture present in the cohort, distinct variation that's present only in Mexico. We created resources such as a variant browser, an imputation panel, and a data sharing platform. And we think this is going to be a very valuable resource for genetic discovery. If you're interested in the MCPS study, you can learn more about it. So we have a data showcase where you can go online and look up the data that's available, the various phenotypes and mortality records, as well as the NMR and other assays that are present in the cohort. And the data is available for researchers. So you can submit an open data access request if there's a disease or a project that you're involved in and you want to work with some of this data. You can submit a project proposal, and currently open data access requests are only available to Mexican-based researchers, right? And so if you want to work with the genetic data, that's going to be through the DNA Nexus platform, where we also have funding to pay for research costs. So right now, you wouldn't have to pay anything to analyze the genetic data if you're a Mexican researcher. If you want to collaborate with us, the MCPS study group, which involves my team in Oxford, or Jaime Berman's team at UNAM in Mexico City, you can reach out to us if there's a project you want to work together with. We're really interested in diabetes and obesidad, so that's our main interest. But feel free to check out the data access policy. Many institutions and funders to thank, and people to acknowledge. So Jonathan Emerson is the person who hired me in Oxford, and he leads the group there. But then we have Professor Jaime Berman and Professor Jesus Alegre Diaz. Jesus has been involved in the study for 20 years, and he was the one that led the recruitment. Him and his team knocked on 100,000 doors in Coyoacán and Iztapalapa. Some of his team have been involved for 20 years, like Raul in the middle. And so they've been very dedicated, a very impressive group of researchers. And we're, of course, very thankful to the 150,000 participants in Mexico City that agreed to be part of the study. So thank you for your time. Happy to answer questions.
Thank you, Jason. Amazing talk. So questions?
Thank you very impressive data, and I'm sure you are really busy analyzing all the variations that causes mutations and all this. But what about the synonymous variants? Because you also found
synonymous variants.
Yes, you found a lot of them, and
most of the variants were synonymous. Yeah, of the coding variant or the exome variants.
Yes. And so, anything you have any analysis on them and what they could mean in terms of diabetes and obesity? Or
we haven't really delved into that, right? So, you know, we characterize the distribution of these variants in this genetic variant profile, and for the rare variant tests, we only looked at the predicted loss of function variant. So we explicitly did not look at the synonymous variants, but there could be effects there. You know, if there's more complex interactions, if there's some type of regulatory effects that are, you know, captured by non-synonymous or synonymous variants, we haven't explicitly looked at that. We have done genome-wide association studies using imputed data, and most, you know, associations for common complex traits are non-coding, right? But yeah, we haven't really delved into synonymous variation.
Yeah, and my second question is now how do you find this, for instance, this mutation in the MAPK315 in the families? I mean, is it those families are really protected, and to what level?
So yes, the odds ratio was about 0.7, so there's about a 30% lower odds of having diabetes if you were a carrier for that variant. I don't know the extent to which that protein truncating variants in that gene were shared among family members. But we do know that there are a higher number of homozygous loss-of-function variants in MCPS in general, right? Because of the family networks, it's more common that you'll have two people within the same family sharing these rare loss-of-function variants, and then, you know, with enough relationships, the likelihood of having two copies, right, inherited from a common relative is more common in Mexico than in UK Biobank.
Okay, thank you very much.
Yeah.
Hi, thank you for the fantastic talk. I was wondering, I mean, with such a diverse genetic variation in the Mexican population, how can we explain, I mean, the Mexican population behave very similar in terms of developing diseases like the obesity? I mean, are you considering like a epigenetic component?
Yes, yeah, no, that's actually really interesting, really thought there, because I mean, for most common complex diseases, it's thought that most populations share the same risk variance. And in fact, in the GWAS Manhattan plot, if you look at the top associations here, like the most significant association is KCNQ1, right? This was an association first discovered in East Asians, like over 10, 12 years ago. But then you also have TCF7L2, which is the top association in European populations, ADCY5. So many of these variants are shared across populations, but then you have a subset of variants that are more common among, you know, specific populations like SLC1611, which is very common in Mexico. Most of these associations, though, are non-coding, right? That's why we like the rare variants, the exome data, because that directly implicates a gene. But with, you know, GWAS, you find mostly non-coding variants, so there's likely some type of regulatory effect. And that involves potential molecular or epigenetic and molecular epigenomic differences, methylation, chromatin accessibility, histone post-translational modifications. And most of the molecular epigenomic data available has been ascertained in populations of European descent. So we're starting to appreciate more and more that there could be, you know, considerable molecular epigenomic differences between ancestries, including in Mexico. Right? There was a study that was published recently using the TopMed data looking at gene expression and identifying variants that associated with differences in gene expression between African Americans, Mexican Americans, and Puerto Ricans. And they found that there were a lot of variants that regulated gene expression that were shared, but there were a subset that were specific to Mexican Americans, right? And it could be the case that some of these signals here, right, that are population specific might have molecular epigenomic signatures that are also unique to these populations, but the disparity of representation in epigenomic data is even worse for non-Europeans there than in GWAS. So I think there's going to be a lot of useful insights coming up in upcoming years, but we still don't know for sure the extent to which, you know, population-specific differences are responsible for that.
Right. Can
Sure.
And my second question maybe is too naive, but what is the final goal of this type of studies in terms of the Mexican population, to try to find like biomarkers?
There's a whole range of research avenues, right, that the genetic data allows us to explore, right? Just by looking at the variation in the exome genome sequencing and genotyping data, you can learn about variation in the population, tells you about demographic events. There's many interesting questions about population genetics and evolution one could explore with the data. I'm in a Department of Population Health, so we care about risk factors for disease. And that's our main interest, right? Most of the research done to this point in MCPS has been looking at risk factors like, you know, smoking, alcohol, adiposity, you know, BMI. But genetics, right, are also risk factors too, right? And if SLC1611 has an odds ratio of 1.3, that's comparable to other exposures. And so it's important to know that if you care about population health, right? And that might help us understand at least part of why there's such a high burden of disease. But when you do analysis like heritability estimation, that tells you the extent to which differences between people in their diabetes risk are due to genetics versus the environment. And I didn't talk about this today, but the heritability for type 2 diabetes in Mexico is no more than 31%, which means that about 70% of the differences between individuals in Mexico are due to environmental factors. So it's likely diet, exercise, or lack thereof. So that's where the genetics would be helpful too. There's, of course, using the rare variants to identify potential drug targets. That could be useful. But biomarker detection, prediction, who's more likely to develop diabetes. And I can talk about another method called Mendelian randomization, but that's a long conversation. So maybe later.
Thank you. Thank you very much.
¿Alguien más? No sean tímidos, ¿no? ¿Nadie? Bueno, ya pasamos el tiempo. Muchísimas. Perdóname, Takuya.
Oh, sorry, Lucian.
Maybe I missed the first section, but the 140,000 individuals are patients of some disease or healthy
individuals? It was a prospective study. Prospective study, which means that when Jesus was recruiting people, it was anybody who was willing to participate. So there wasn't ascertainment for any particular disease, right? It was, basically, would you consent to be part of the study, share your results? And that's basically what a prospective study is. You recruit as many people as possible from a representative population, find out as much information as possible, and then wait to see over time who dies of specific diseases so you can link back what the major causes were.
So there's some information of phenotypes, not only genotypes.
Oh, absolutely. Yeah.
Is all of information published?
Yeah. You can check out this link here, the one on the top right.
Okay.
This has all the phenotypes present in the cohort. Everything we have currently is there for public access.
But individual information is sacred.
Yes, it's anonymous. It's anonymous information. Yes, so we have anonymized IDs on there. Yeah, but it's linked with the genetic data. So if you wanted to do a genetic analysis of a type of cancer or immunological disease, you can see what the case numbers are and how many people carry risk variants. So, yeah.
Okay, thank you.
Maybe last one, la última, creo.
Very nice talk. One of the leading causes of mortality in Mexico, including Mexico City, is violent death.
Is it which one?
Being killed by someone else. Do you think there could be a genetic association with that?
It's a controversial question. It really is, because there's a ton of confounding, right, with socioeconomic factors, with environmental factors. So it's very difficult, even if you find a genetic association, to disentangle that as a causal effect, right, that the variant is directly causing the violence versus something that is confounded by environmental factors, such as, higher crime in the area in general, lack of access to resources, poor policing, whatever it may be. It's very difficult to. You can find an association, I'm sure, but one has to be very careful when trying to link causal pathways from genetic variants to these outcomes, especially when you deal with issues related to cognitive, behavioral outcomes, which is why you got to be careful when you study intelligence or educational attainment, because there are a lot of environmental confounders that could underlie genetic associations there. But it's an interesting idea. I mean, I won't say any more now, but I'll think about that. Thank you.
Ok, muchísimas gracias a todos por venir. Thank you, Jason.
Sí, gracias. Mucho gusto.